So welcome, everyone. My name is Deb Rachko, and I am the president of the Kennisis Lake Cottage Owners Association. Thanks for joining our first What's Up Kennisis webinar. Last year, the KLCOA conducted a general survey of the membership to help guide and focus the board of directors with planning and direction. A lot of great information came out of that survey. One of those things was the desire of the membership for more education on a broad range of topics. Another was the importance of maintaining good water quality, and specifically, the number one concern was algae. Putting all of those things together brings us to our first educational webinar tonight, entitled Our Water Quality and Blue-Green Algae, What You Need to Know. I'd like to now introduce KLCOA Vice President and Stewardship Director Jim Prince, who will facilitate tonight's meeting, tonight's webinar, and introduce our guest speaker. Enjoy the presentation. So, Jim?
Thanks, Deb. Good evening, folks. I am unmuted, yes. Good evening, folks. Happy that you could take a couple minutes out of your beautiful Wednesday evening to join us tonight. Tonight's webinar is about our water quality. And if you remember what we did in the spring meeting, we talked a little bit about how our survey results are forming what we're going to be doing in terms of programming for the KLCOA. So one of the questions we asked, what are the particular concerns about water quality in our lakes? And algae was the top response. So nearly 70% of folks did. So tonight we've got a talk from Liz, Dr. Liz Favot on blue-green algae. So we've done our welcome. 7:10, Liz is going to take over. I'm going to give a bit of a Kennisis backgrounder about our specific water quality. And then please put your questions in chat, and we'll see what happens near the end of the webinar, if we can actually open it up and unmute people. I'm not sure if I can get to that control part. And we'll answer those questions and we'll adjourn. Okay, so the Kennisis Lakes we've been doing water quality monitoring in a variety of ways in partnership with the provincial government and with two provincial government programs, but one now is run by FOCA. One's called the Lake Partner Program, and the other one is the Ministry of Environment and Conservation and Parks. It changes the name all the time. For the Lake Partner Program, on the far right hand, you can see we do samples and test for 4 things. We do that once a year in May, and our citizen scientists on KLCOA do that. So 2 basins on the Big Lake and 1 on the Little Lake, and we have that data quite reliably for nearly 20 years. Few breaks in the data was breaking COVID, and a couple of years people just weren't able to get out, but it's quite good. We are also a lake trout lake, and we are recognized as a lake trout lake. And because of that, we have the benefit of the ministry coming out and doing a real deep dive in terms of parameters. And they do that, unfortunately, only every 5 to 8 years. But they do it in 4 basins, 2 in Big K, 2 in Little K. And the timing was around 2002, 08, 15, and 16. And 16, I'll tell you why there's an exclamation mark there. And they take a broad sampling approach, but primarily around lake trout health. And a lot of Haliburton lakes actually are benefit from that testing program. So in 2015, the MECP came, a fellow named Dana Cruikshank, and he found quite a large spike in the total phosphorus and some nitrogen and ammonia compounds in the lake. The green line that just came up shows what's actually happening to our lake. So the blue bar graph charts are the Big Lake, and the red ones are the Little Lake. And you can see over the years that, in fact, we are increasing in total phosphorus, and that is, we don't have agriculture. We desperately hope people aren't fertilizing their lawns. So most of it is a little bit climate change, but probably most of it are nutrients from our septic systems, almost entirely. And over the years, they have increased. And if you look at the line across the top here, 10 micrograms per liter total phosphorus is considered a reasonable measure for a healthy lake. So our lakes are healthy, but we are trending in the wrong direction. So in 2015, when the MECP came and we saw this spike in Big and Little K, 2015 specifically, we were alerted to the fact that we are likely having some septage problems in both lakes and in particular in Little K. The ministry, Dana got permission to come back the next year and found a slight reduction in Little K and Big K getting more or less back to normal, but both still relatively high. And in the next slide, I'll tell you a little bit about how we responded to that. But in 2020, there was a blue-green algae event in Little Kennisis. It wasn't confirmed by the MECP, but we sent photographs. A couple of blue-green algae folks looked at it and said, yep, that's a small event. And it dispersed naturally in a couple of days. So it wasn't too serious. If you were watching your e-blasts, we reported it and cautioned folks. And that's the last known event we've had, and one of the first events we've had, we believe. Okay, so just to a little bit more background on perhaps why we're seeing that effect in Little K. These are the images of the lakes, and Little K is much smaller than Big K, but here's some real numbers. So Big K has 760 properties, Little K has about 280. And if you look at the surface area differences and the lake volumes, I've summarized it in the bottom on the right-hand side. Big K versus Little K. Big K has 2.7 times the number of properties, but Big K has 6 times the surface area and 9.5 times the water volume. So clearly there's a dilution benefit happening, but Little K in a correct terminology is at capacity, and we really think it's over capacity. So this is a signal that in particular everyone, but in particular the folks on Little K really ought to be certainly focusing on septic system health and shoreline naturalization to help take some of those nutrients out. In the Big Lake as well, but the way we're trending on Little K, it's particularly important in Little K. So what did we do in response to that? Because we knew the MECP wouldn't come for 5 to 8 years again. We were going to continue with lake partners. So in 2017, the KLCOA board approved a limited water quality testing program of around $2,000. There wasn't a Halliburton-wide protocol that we could look at. There were other protocols around. We did get help from the ministry staff. We got help from the good folks up at the Dorset lab who do the analysis for the Lake Partner program. And CHA had a water quality workshop around that year, which was very beneficial. And it kind of got us over the hump to say, yes, we can do this. So we implemented a citizen science test program. We go out twice a year. We do 5 basins, and the image of the map shows you where those 5 basins are. We've added Paddy's Bay in because we think it's at risk. We're testing for a subset of that MECP, which is dissolved oxygen and temperature, which gives you an idea what's happening in terms of oxygen levels in the lake. Again, total phosphorus, and then the ammonia nitrates and nitrites. But it has left us with a lot of questions. We're citizen scientists. We're not limnologists or water quality folks like you're going to hear from Liz tonight. Are we sampling for the correct indicators of lake health? What's the best timing for the sampling? Are our test protocols acceptable? And how do we analyze and present the data, in particular to the members of our association? And where do we store that so other people can look at it and trend? So with that gives you a picture of where we're at and the fact that we have had a blue-green algae event. And next, I'd just like to introduce, I'm going to go back here. I've got Liz's CV, and I'm going to have to minimize my go. Can you still see my screen? I think so. Okay, now I'd like to introduce Liz, Liz Favot. Liz completed her PhD from Queen's University about 1.5 years ago, which focused on a multi-century trends in water quality and the environmental drivers for recent blue-green algal blooms in minimally human disturbed lakes in Ontario. After Liz's PhD, she lived in Halliburton for about 1 year, where she worked out of the Dorset Environmental Science Center, which I talked about, as FOCA's Assistant Lake Stewardship Coordinator to help to deliver the Ontario Lake Partner Program. Liz currently resides in Sudbury, Ontario, where she works as an aquatic research biologist with the Ministry of Natural Resources to study food web and reproductive ecology of northern fish communities. So welcome aboard, Liz, and I think we can probably hand it over to you to share your screen. I'll stop sharing.
Thanks. Okay, I'll just get my screen up. It says host disabled participant screen sharing.
I can fix that. All panelists. There you go. Should have it now.
Thanks. Yeah. Okay. All right. Can you guys see my slides?
We sure can.
Okay, so yeah, thank you so much for inviting me to speak at your first webinar in the series. Tonight I'm going to be speaking to you not on behalf of any ministry that I've worked for in the past or that I work for now, but I'm going to speak to you as someone who has, for my PhD, as well as currently in some postdoctoral work, researched blue-green algae. So yeah, interestingly, my 9 to 5 is actually entirely focused on fish research now, but on my evenings and weekends, I've been plugging away at a provincial scale blue-green algae study, which I'll talk to you a little bit about this evening. So maybe many of you already know the basics about blue-green algae, but just to make sure everyone's on the same page, we'll start there. So blue-green algae is more correctly called cyanobacteria. They mean the same thing, but the reason the term that blue-green algae is misleading and why the scientific community has abandoned it is that cyanobacteria are actually fundamentally different from other true forms of algae like diatoms and green algae. Rather, cyanobacteria are photosynthetic bacteria. They are one of the oldest organisms on the planet. They have evolved about 3 billion years ago, and they played a critical role in the evolution of nature as we know it because they're responsible for the oxygenation of our atmosphere. Because they have such a long evolutionary history, cyanobacteria are well adapted to many different environmental conditions, and so you can find them in most illuminated environments on Earth, everywhere from ice in the glaciers at the poles, in hot springs, as a part of lichens on rocks, like you see all over the Canadian Shield, and of course, they're a very common component of our oceans and freshwater algae communities. There are over 2,700 named species of cyanobacteria, and the group probably contains several thousand more that we just haven't discovered yet. And the species of cyanobacteria have diverse forms. They can have a unicellular lifestyle, like these picocyanobacteria, or they can be colonial, or groups of cells, like microcystis, to filamentous forms or chains of cells. So that's species like Anabaena, or now called Dolichospermum. And the size difference between some of the smallest cyanobacteria to some of the biggest, like microcystis colonies, is equivalent to the size difference between a fleck of brown cayenne pepper and a soccer ball. And the size difference actually has some ecological implications too, because the larger forms like microcystis colonies are less likely to be eaten by phytoplankton, or zooplankton, sorry. So they're just harder to grab and consume these colonies. And of all the thousands of species of cyanobacteria, there's relatively only a handful that form blooms in fresh water, and those are some of the groups shown in this picture. So my PhD research used paleolimnology, which I'll explain later, to examine long-term environmental conditions and potential drivers of cyanobacterial blooms in Ontario lakes. And then my postdoctoral research characterizes nutrient conditions and spatial and temporal patterns in Ontario lakes with confirmed cyanobacterial bloom reports from the public. So basically, if a member of the public sees something that they suspect is a cyanobacterial bloom in their local water body, they're encouraged to call 1-866-MOE-TIPS to report the bloom. In response to that, a local government official or public health official will come out and take a sample that looks something like this, and then someone at MOE in Etobicoke puts it under a microscope, and if the community is dominated by cyanobacteria, that goes into the record books as a confirmed cyanobacterial bloom report. And so there's a data set of all these confirmed cyanobacterial bloom reports that have occurred across the province of Ontario, and I'm working on looking at where and when these public reports of cyanobacterial blooms came from, as well as what the conditions were like in the water body that they came from. And so I'll talk a little bit about that tonight. So what exactly is a blue-green algal bloom? There's actually no strict scientific definition, which gets really confusing, but it generally describes a visible accumulation of algae. And it's when one or a few species come to dominate the algal community in a water body. So you can have algal blooms that are not comprised of cyanobacteria or blue-green algae. You can have diatom blooms or green algae blooms. But tonight we're going to be specifically focusing on blue-green algae blooms or cyanobacterial blooms. And blooms can be measured in different ways, and that's where it gets confusing in the scientific community, because you can measure a bloom or determine that something is a bloom based on total cell counts of the algae per liter, or based on the proportion of the algal community that's a certain species, or based on concentrations of photosynthetic pigments like chlorophyll A. The name blue-green algae is also kind of misleading because blooms are not always blue-green in color. So if you take a couple seconds, can you guess which of these images are blue-green algae and which are not? So these three images show blooms comprised of cyanobacteria, but this image shows duckweed, which is a plant. This shows green algae, which is more stringy and slimy in texture, and this is pollen on the water. So even to the trained eye, sometimes blue-green algal blooms can be tricky to distinguish from other types of algae blooms until a sample is put under the microscope. But generally, if you see kind of a consistent opaque pea soup green kind of growth, it could be cyanobacteria, in which case I really recommend that if you spot something that you suspect is a blue-green algal bloom, that you call the Spills Action Center at 1-866-MOE-TIPS to report it. Many studies hypothesize, and some global satellite studies are now supporting that cyanobacterial blooms are on the rise globally. And they've been detected in more and more water bodies over the last few decades. And here are just some Canadian headlines about cyanobacterial blooms that have had recent media coverage. This one kind of caught my eye, showing that research is now showing that blue-green algal blooms on Lake of the Woods are now lasting longer. And this has made big headlines recently too, which is the blue-green algal blooms on Lake Superior, which is a very low nutrient, low production, cold system that we think prior to the changing climate did not support frequent cyanobacterial blooms. So in Ontario specifically, Jenny Winter and colleagues published a study in 2011 showing an increasing number over time, so these are years here, of confirmed cyanobacterial bloom reports. So these are the number of reports over time. And so I'm looking at this data set a decade later to kind of update what's the state of cyanobacterial bloom reports in Ontario now. And what we've found is that they've continued to rise dramatically over the last decade. There's a couple important points about this data set to make note of that might have impacted the number of confirmed reports. Firstly, the provincial response protocol was initiated at this blue vertical line here in 2005. So that might have increased public awareness and reporting in general. So that might have been why we see this initial uptick in reports at that time. And then secondly, there was a transition in program analysts, the only one that's occurred over the 20 years in 2013 and 2014, which might account for lower reporting in those 2 years. But generally, we're seeing that the number of reports of blue-green algae blooms are increasing over the last 2 decades in Ontario. And then on this bottom panel here, we've looked at the number of water bodies reporting a blue-green algal bloom for the first time. So we're also seeing growth in the number of water bodies with blue-green algal blooms over time. These are the locations across the province where confirmed blue-green algal bloom reports have come from. We find that there's a relationship between population dense and accessible areas, or basically the major road network within Ontario, and where the confirmed bloom reports come from. This makes sense. The data set starts with a public complaint or a member of the public suspecting that there's a blue-green algal bloom. So the bloom are only going to come from where there's people to observe them, which is why there's so few in the northern portion of the province. It's not that cyanobacterial blooms don't occur in the north. We know that they do. But this data set is based on public reporting. And so that's why we have no data points from the far north. So what we found is that there's a large prevalence of bloom reports coming from the Georgian Bay ecoregion. 51% of all reports came from the Georgian Bay ecoregion. And we think this is because it's a major intersection of people and lakes. So it's an area that's more populated than further north, but compared to further south, which is even more populated, it has more lakes and more people recreating on lakes than the more urban south portion of the province. So we think this is why most reports are coming from the Georgian Bay ecoregion. And now, although nutrient pollution from runoff is the most common culprit of cyanobacterial blooms globally, we, in this study, combined the reports of confirmed cyanobacteria with Ontario Lake Partner Program data to try to understand the nutrient conditions of the lakes where bloom reports are coming from. And what we found is that 41% of the bloom reports in Ontario came from lakes with spring total phosphorus less than 10 micrograms per liter, which is like a very low range. We call that when TP is less than 10 micrograms per liter, we call that an oligotrophic or low production lake. And that's up from 25% in oligotrophic lakes, which was reported by the initial study on this data set 10 years ago. We also found, and you can see that this on this graph here, by looking at where the majority of these shapes lie compared to this red line for the provincial water quality objective, we found that 89% of lakes with confirmed cyanobacterial blooms have spring TP that's within the provincial water quality objective of 20 micrograms per liter. And then lastly, we found if we just focus on the Georgian Bay ecoregion here, and then what we're looking at is the teal color is lakes with no confirmed bloom reports. The yellow color is lakes with 1 or 2 confirmed blooms, and the red color is lakes with 3 or more confirmed blooms. And we found that in the Georgian Bay ecoregion, the y-axis here is spring TP, and the black dot represents the mean of each of these groups. We found that there was no significant difference in spring total phosphorus in the Georgian Bay ecoregion in lakes that had no blooms and lakes that might have had 1 or 2 confirmed blooms. And so what this suggests is that, yes, nutrients are critically important to fuel blooms, but they're not the whole story. There's other factors aside from simple nutrient runoff that can drive blooms, especially in areas like the Georgian Bay ecoregion, where we have relatively low nutrient systems and relatively pristine water bodies. One of the major consequences of cyanobacterial blooms that people are most concerned about is their ability to produce toxins. There are 4 main groups of toxins produced by freshwater cyanobacteria, shown here. And then there's hundreds of variants within each of these categories of toxins. And we haven't discovered them all yet. But by far the most common toxin associated with freshwater bloom, microcystins, which are toxic to the liver. And we do have standards in place in Canada for the level of microcystins, both in drinking water as well as in recreational water. But we don't yet have guidelines for some of the other toxins. But we haven't detected them as much as we've detected microcystins associated with toxic blooms in Canada. But it's not all about the potential for toxin production. There are a myriad of negative consequences, both of toxin producing and non-toxic blooms. So of course, blooms that produce toxins are a health risk to humans and animals, but whether or not blooms are toxic, the biomass produced during blooms reduces water clarity or increases turbidity. And this can lead to biodiversity loss through a reduction in the growth of other photosynthetic aquatic organisms. And can also, this reduced water clarity can reduce the aesthetic and economic value of a water body. Bottom water oxygen depletion associated with the end of blooms when excess organic material is being decomposed can reduce the suitable habitat available for certain aquatic organisms and result in fish kills. And bottom water oxygen depletion can also exacerbate internal nutrient loading in the sediments, which then results in a kind of positive feedback cycle that can further bolster future blooms. The causes of algal blooms can be highly complex. A lot of current research is focused on being able to predict the conditions under which blooms will occur in the future. But there's a line in this review of blooms in Canadian waters that I really like that states that, let alone predicting future blooms, it's actually difficult enough for us to even tease apart precisely which factors or thresholds are surpassed to have caused past blooms in certain water bodies. We're still kind of working on that a lot of the time. Despite the complexity of causal factors of blooms, you can conceptually organize the interacting factors that lead to blooms into these three categories. You have elevated nutrients, both from natural and human-caused sources. So nutrients can come from runoff from the catchment, or they can come from internal lake sources or the mud at the bottom. Then we have specific weather conditions, like especially ones that lead to warm or stagnant water, or also extreme precipitation events that might lead to nutrient pulses or excess nutrient runoff. And then there's also food web alterations or changes in basically how much algae is being consumed by upper food web organisms. Climate change can alter conditions in each of these categories to make conditions more favorable for blooms to occur. So you can kind of think about it as a threat multiplier in the case of conditions that promote cyanobacterial blooms. Switching gears a little bit here, before I describe some of the main ways that climate warming is making blue-green algal blooms worse, we need to understand the process of thermal stratification in lakes. So maybe many of you already know this, but maybe it'll be a refresher. So in the spring, after the ice melts off the lake, water is cool and uniform of about 4 degrees Celsius all the way from the top to the bottom of the lake. And so all the water in the lake mixes together and oxygen and nutrients are spread throughout the water column. Through the summer, the surface waters of the lake heat up, and this creates layers of water of different temperature and therefore different density. And if the thermal gradient is steep enough, or if the temperature differences are big enough, the layers of water become too different in density to mix with each other. And this is why if you jump into a deep lake near the end of the summer, if you reach deep down with your feet several meters, you might hit a layer of much colder underlying water. And we call this layering of water of different temperatures through the summer thermal stratification. And during stratified conditions, the hypolimnion, or the cold bottom waters, are isolated from atmospheric oxygen. And so the oxygen in this layer that's there in the spring is, once it becomes isolated through thermal stratification, is slowly depleted throughout the summer as organisms within it consume the oxygen. And when oxygen is depleted from the bottom waters, nutrients bound up in the sediments can be released into the overlying waters, and this process is called internal nutrient loading. And so it's been widely hypothesized that climate change will result in more frequent, severe, and more widespread blue-green algal blooms on a global scale. And this is because cyanobacteria possess certain adaptations that allow them to thrive and outcompete other algae under the conditions imposed by climate change. So specifically, if we're looking at a typical lake in this area of the world, so moderately deep temperate lakes, recent warming is resulting in longer and stronger periods of thermal stratification in lakes during the summer, as well as a longer ice-free growing season. And bloom-forming cyanobacteria do well under these climatic conditions, firstly because they generally grow better than other types of algae at water temperatures around 25 degrees Celsius and in excess of that. So they do well in really warm surface waters. Secondly, many bloom-forming species of cyanobacteria possess gas vesicles, which other types of algae don't have. And this allows them to control their buoyancy. So it's kind of like a little life jacket. And it means that unlike other algae groups, like the diatoms, for example, that slowly sink out of the water column and slowly sink out of the area of light that they need to photosynthesize when the water column is stagnant, like under thermally stratified conditions, cyanobacteria can actually optimize their position in the water column and are not reliant on turbulence to remain suspended in the upper waters. where they can photosynthesize. This buoyancy regulation also allows them to monopolize the illuminated areas of the lake and outcompete other types of algae like diatoms by shading. And the buoyancy regulation also allows species of cyanobacteria to migrate down up to about 10 or 15 meters, we think, to access bottom water nutrient pools, which can form through internal loading from the sediments. So the main question that I examined in my PhD research was what environmental conditions have changed in lakes that could be driving recent blooms. And since the lakes that I studied have very little historical monitoring data, I used paleolimnology to answer this question. And so paleolimnology is a branch of lake science that examines fossil material in lake sediments to determine the environmental history of an ecosystem. So how this works is that material from the atmosphere, from the catchment, as well as from within the lake, are continually deposited as sediment to the bottom of the lake. And we take a core of the sediment that looks something like this, which then provides us a continuous record of environmental conditions in and around the lake from present day at the top of the core, going back centuries or millennia as you move deeper in the mud. And so this allows us to extend the monitoring window for a particular lake, which as Jim mentioned for Kennisis might be 20 years. We can extend it back centuries or millennia with paleolimnology to establish what the lake was like before major disturbances of the industrial era. And so Rachel Carson said in her book, The Sea Around Us, the sediments reflect all that has happened in the waters above them and on the surrounding lands, if we're wise enough to interpret the signals that they contain. And so some of the materials that preserve well in the sediment that we can use to interpret environmental change include chlorophyll A, which is a photosynthetic pigment in all plants and algae. So it can be used to infer whole lake production through time. Cyanobacterial actinides, which are resting cysts of certain types of bloom-forming cyanobacteria. Diatoms, which are another group of algae, and they have a glass or siliceous cell walls that allow them to preserve for thousands of years. And we can use them to interpret how nutrient levels in a lake have changed because different species have different preferences for nutrient conditions. And we can also use diatoms to examine how climate warming might be impacting a given lake, because longer and stronger thermal stratification that I was talking about before should favor slower sinking species of diatoms. Chironomids or non-biting midges, these are a group of flies that have a larval stage that lives in aquatic environments in the mud of lakes. And they're sensitive indicators to changes in oxygen near the sediments. And they can also be used to indicate changes in temperature. And then lastly, the cladocerans, which are an important grazer on the algal community, and some species of Cladocera are more efficient grazers than others, or more efficient at eating algae. And so we can look at shifts in their fossil communities through time to indicate changes in food webs that control how much algae there is. So to demonstrate how paleolimnology works and how we establish the environmental history of a particular water body and how that can help us determine the drivers for current blue-green algal blooms, I'll tell you about the research I did on Calendar Bay of Lake Nipissing. The municipality of Calendar actually pulls its drinking water from Calendar Bay for 4,000 people. And so toxin-producing cyanobacterial blooms that have occurred in the bay almost annually over the last 10 years are exceptionally concerning here. So Calendar Bay, beginning in the late 1800s, it was a site of clear-cut logging. There was a tourism boom to the depressed lumber town in the 40s because of the world's first surviving quintuplets that were born here. Since about the 1970s, the logging industry has drastically declined, but cottaging tourism and the walleye sport fishery are really important contributors to the local economy. And cyanobacterial blooms have only been observed in Calendar Bay since 2000. But what about on a longer time scale? What was Calendar Bay like before the Industrial Revolution and substantial human impacts? Were there cyanobacterial blooms in the bay in the 1600s or the 1800s that went unnoticed through the 20th century? Or are the frequent intense cyanobacterial blooms that we've seen in the bay over the last 20 years truly a new occurrence here? And if they are a new occurrence happening so frequently like that, then what has changed in the ecosystem of the bay to cause that to occur? So I went out and collected some sediment cores from my PhD to try to figure some of these things out. And so this is the change in the diatom community of that sediment core over time. So how you interpret these graphs is each of these panels is a different diatom species. And as you move down the page, you're moving deeper in the sediment core. And these are the years here associated with each layer of sediment. And then the length of the bar represents the amount of that species that there was at that time period. So what we saw was that in the pre-disturbance, or in the 1800s and earlier, the community was dominated by Algae, or this type of diatom. Then we move into a distinct period in the diatom fossil community from about 1890 to 1970. And during this time, there was clear-cut logging in the catchment. And what we see is about a 40% decline in the abundance of the previously dominant Algae group, and their replacement by Fragillaria mesolepta, which is a diatom that prefers higher nutrient conditions compared to Algae species. So that suggests that there were higher nutrient levels in the bay during this period. Then we move into sediments from the 21st century, and this is also the period where we've observed cyanobacterial blooms in the bay. And what we saw was a decline in Fragillaria mesolepta, that nutrient-loving diatom, and their replacement by an increase in these very small, slow-sinking diatom species. And this shift from the pre-disturbance period dominated by Algae species to a period coincident with the observed blooms over the last 2 decades, in which these small, slowly sinking diatoms are at higher abundances, suggests to us that longer or stronger thermal stratification has occurred in Calendar Bay over the last couple of decades. And as I mentioned earlier in the presentation, this same enhanced thermal stratification predicted to favor this diatom shift that we saw also benefits cyanobacteria because they can regulate the buoyancy to optimize their position in the water column under these conditions. And so we think that this signal in the diatom record points to climate-mediated changes in lake thermal stratification, an important driver of blooms in Calendar Bay. Large shifts also occurred in the chironomid assemblages of Calendar Bay, and this is how we find out about oxygen. But I'll skip over these quickly and just get to the summary. But basically what we saw is that the pre-disturbance period was dominated by Procladius, and then it switched to a chironomus. And these are what people call bloodworms. They're red in color because they have a type of hemoglobin to like we have in our blood, which allows them to very efficiently bind oxygen. And so they can actually withstand periods of really low oxygen compared to some other species of chironomids. And we saw those increase in abundance as we move towards more recent times. But so in this diagram, we have time running along the x-axis. And the green shaded area represents the period when we saw cyanobacterial blooms in Calendar Bay. And the top panel of this figure displays the concentration of photosynthetic pigments in the core over time. And so what we saw was an increase in algal production from about 1930 to present day. In terms of climate records, these are from the North Bay Climate Station. What we saw was an accelerated warming in air temperature since about 1980, declines in wind speeds, which can lead to more stagnant conditions in a water body, less turbulence, fewer days of ice cover per year throughout the record, and wetter conditions in the 21st century. And these climate parameters are all trending in a direction that can favor blue-green algal blooms. And then the bottom 2 panels here show reconstructed bottom water oxygen from chironomid assemblages and reconstructed total phosphorus trends from diatom assemblages. And these trends show especially low bottom water oxygen over the last decade, indicating a greater potential for internal nutrient loading. And diatom inferred total phosphorus indicates that conditions were about 13 micrograms per liter in the pre-disturbance period, underwent a period of nutrient enrichment through the 20th century, and has recovered somewhat from eutrophication over the last 20 years during the period that blooms have occurred. And another important aspect of paleontological studies is that they allow us to quantify what conditions were like before direct monitoring and human disturbance began. So for example, here, if we take an average of these paleo inferences for oxygen and phosphorus from about 1850 and before that, we can actually provide historically informed management targets for this lake specifically. So according to before humans really started changing the system, oxygen should be around, and this is bottom water, end of summer oxygen should be around 2 to 2.25 milligrams per liter, and phosphorus should be around 13 micrograms per liter. So to conclude, for this Calendar Bay case study, compared to the pre-disturbance conditions in the 1800s, the 21st century in which frequent blooms have been observed is characterized by stable or even declining nutrient concentrations, which suggests that other factors are also at play in driving the blooms. We also see an increase in diatoms indicating periods of longer or stronger thermal stratification in Calendar Bay during the period of blooms. And we see reduced bottom water oxygen at the same time. So it seems plausible here that with climate warming, Calendar Bay is transitioning from a well-mixed bay of water to one that's more consistently thermally stratified. And under these conditions, oxygen would be quickly lost from the bottom waters, and internal loading could create deep water nutrient pools to fuel algal blooms in the bay that we've seen in recent decades. And now if I consider the findings from my PhD research as well as my postdoctoral research, sorry if you can hear my parents' puppy, by the way. What we found is that nutrients are critically important to support blooms, but in terms of the drivers or triggers for blue-green algal blooms, they're not actually the whole story. And we found that our inferences and reconstructions from sediment records indicate that background nutrient conditions seen several centuries ago are in some cases, that's how the lakes still are today. So nutrients haven't increased in some of these systems, yet blooms are occurring in the modern climate. And this is likely because climate warming has several consequences that bolster blue-green algal blooms, especially through a longer ice-free growing season, warmer surface waters, longer and stronger thermal stratification, and exacerbated internal nutrient loading. For what I can recommend for management, climate is hard to control. We need to look at what's within our sphere of influence. The science in what triggers blooms that I've outlined here is pretty complex, but the implications and what we can do to mitigate blooms is actually relatively simple. What can we control? We can control nutrient inputs. We need to keep nutrient concentrations in lakes as natural as possible. And what specific level that is in each lake can be determined through paleo studies like the ones I've done, or through long-term monitoring. So if you have at least 5 years of monitoring data. Controlling blooms once environmental conditions are right for them to reoccur annually is very, very difficult because it sets up positive feedback cycles in the system that allow these blooms to reoccur year after year. Chemical and other in-lake treatments to try to get rid of blue-green algal blooms are expensive, and they're not a viable long-term solution. The best thing we can do is try to prevent blooms from occurring in the first place. And one of the best ways we can do that is to minimize nutrient pollution running off from human activities in the catchment. And so a very easy way to do that is to intercept nutrients flowing off the land with terrestrial plants, which can sequester these nutrients. So I highly recommend keeping shorelines and catchments as natural as possible and as forested as possible. There's always going to be ecological surprises since there are so many interacting factors that can lead to blooms. But I do believe that with further research, we'll be able to pinpoint specific lakes that might be most sensitive to developing algal blooms. And maybe a good start to that is to look towards lakes that have moderate nutrient levels or are classified as mesotrophic with 10 to 20 micrograms per liter phosphorus, and lakes that are relatively shallow so that low oxygen conditions develop rapidly with stratification. But I should mention, back to the provincial scale study of blue-green algal blooms I did, we found blooms occurring in lakes down to, with spring total phosphorus down to 3 micrograms per liter. So basically blooms can happen anywhere at any nutrient status. Lastly, I've found that bottom water oxygen and nutrient concentrations during stratified periods that indicate the occurrence of internal nutrient loading might be a more useful parameter for determining the potential for blooms rather than surface water concentrations. So for groups that have the means, I think we should incorporate oxygen profiles at multiple times of the year, and maybe a stratified period bottom water phosphorus sample into our monitoring programs. While it takes a lot of specialized work and time, understanding environmental histories is the key to putting current water quality issues into context. We need to establish baselines to know what we should be managing to. And when monitoring data don't exist to do this, there's always paleolimnology and sediment records, which are a useful approach to be able to open up the window into the past and see what lakes were like prior to disturbance. So that was really probably way more detailed information that you wanted to learn about cyanobacteria on a Wednesday night, but here are some take-home messages for you. So cyanobacteria are among the most ancient organisms on Earth, and when they're in balance with the rest of the ecosystem, they're an essential and beneficial component. But blooms or excessive growths of certain species of cyanobacteria can have negative ecosystem level impacts. And unfortunately, we do expect that with continued climate change, blue-green algal blooms will likely become increasingly frequent and severe. And society and lake users and managers need to be prepared and have plans in place in order to minimize the negative impacts of these events. And that includes minimizing nutrient enrichment, starting up detailed monitoring programs, I think is a great step, as well as increasing awareness and reporting of these events so we can really understand what the environmental triggers are. Thanks for listening.
Thank you, Liz. That was fantastic. That was a great overview and some tips and ideas for folks that have waterfront properties to manage some of the nutrient input into the lake. Deb was going to have a look at chat coming in and see if we've got some questions. I don't know if anyone has.
So far, we do not have any questions. So if people want to submit those now, that would be great.
Yeah, or I have the ability to unmute folks. So if you wanted to put your hand up, you could raise your hand on the screen. I can allow you to talk and we could just have the questions verbally as well if you didn't feel like typing in a chat. I see some folks on the list who I know have questions.
Oh, okay, we have a question in via chat. Thank you, Liz. You did a great job of explaining blue-green algae for all of us. Sorry, that's not a question, that's a compliment to you. And that was from Elizabeth DeMarsh.
Okay, we just give it a second. I don't see anything else.
No.
Okay, we're running well on time here. I've got a question for you, Liz. Sure. I'll use Kennisis Lake as an example. So we did have the small event in Little Kennisis. We did report to the MECP. There was no sample taken. Do you know what kind of report that would be recorded as?
I don't, and I will look into that for this, because we're getting ready to submit this research in a couple of weeks to be published. But that's really interesting because something I think a lot about is it's like there's so many blooms that go unreported, but that's another instance where it was reported, but if a sample wasn't taken, then I don't think it would have ended up in the data set that I'm working with. But maybe it's tracked elsewhere.
So we did the hotline report called Chris Johnson. I took a sample, held the sample, and unfortunately that was November 2020 when we had 11 blooms in Haliburton County.
Yeah.
And the MECP weren't ready for that, so they couldn't respond. I think if we had been the only one, he probably would have come straight to Little K and done a sample, and we would have been on the record. So that's another factor which reflects what you're saying, that it was 30 degrees in November. It was dead calm for nearly a week. And the conditions were perfect as you've described for something to happen in terms of a release from the bottom. So,
Yeah.
Okay, and I do have a couple more questions coming in. So I have one from Julie. Thank you, Liz. Question: can you talk more about what precautions we should take when recreating on Lake Kennisis when a bloom is suspected?
That's a good question. And I'm not like a public health official, but I think generally the guidance is if you see a bloom, you shouldn't be going in the water, but as long as there's no visible accumulation, you're probably still good to swim. Ingesting the water is obviously completely off limits if you suspect any type of blue-green algal bloom. So precautions, I think just not swimming if you see visible accumulations. An interesting question would be like, should you shower with that water? Because I have a cottage too, and I pull water from Lake Nipissing, which has blue-green algal issues sometimes. I probably, like, out of an abundance of caution, I don't think you should be putting the water on your body if there's a visible accumulation of blue-green algae. So
I'm going to add to that question because I know when we did have the appearance of one, people asked me. So the bloom was found on Little K. How big of an area are you concerned about if one spotted? So you're on Little K. Should people on Big K be concerned as well?
Yeah, I'd say if you were on the other lake, as long as you don't see anything in the water, you're probably okay. But actually, unfortunately, like, we don't actually know that for sure. And there's been instances where toxins can actually be higher in the water after a bloom dissipates. So after you can't see anything in the water, which is really kind of scary. So I would just take an abundance of caution in the couple of weeks following a bloom. But probably if you're on a separate lake and you don't see anything growing in the water on that lake, you would probably be okay. But I guess this is all just like good evidence that we need better policies surrounding blue-green algal blooms and public health precautions, especially if we think they're going to be an increasing feature on the landscape in the future.
Yeah. And one additional comment on that, in Muskoka, when there's a blue-green algal bloom, the Muskoka Health Unit has an information process that they actually do an alert for people on that lake, a formal alert. We don't have that in Haliburton at all. We don't have that in the Kawartha Haliburton Health District. We're lobbying for that now because the only way that folks in the KLCOA or Kennisis Lake community would know is if we alerted them. The Dysart doesn't do it. Haliburton County doesn't do it, although we have formally been reporting to Dysart, Haliburton County, and the health unit to perk their interest in trying to get involved. So it will evolve. It will have to evolve because this is people's health and their pets' health are at stake as well.
Yeah.
Okay, so there are a couple more coming in. In terms of measuring oxygen and nutrient levels, is the purpose to gain an understanding of how this changes over time and how it correlates with blue-green algae blooms, or is it also to use this data to then put preventative mitigating measures in place to assist in lessening or preventing the blooms?
That's a good question. I think both. I think absolutely both. Yeah. Yeah, because I think we should be, it's a good indicator of things that are changing that might cause blooms, but also if we monitor them over the long term, we should look at what those conditions were like when there weren't blooms in the lake and try to manage to those conditions if they're changing. So.
Okay. I've got another one here. How effective are filters in removing toxins from lake-sourced drinking water?
My understanding is that they're not at all. And especially this isn't a filter, but boiling water actually makes more toxins in the water because it breaks up the cells and so the toxins come out of the cells. So yeah, really you should not be drinking the water if there's been a bloom in the last couple of weeks.
Okay, and when we tested the bloom in November, and Jim, I'm going to let you say that. I'm just reading it as it is. When we tested the bloom in November, did you test all over Little K? Were the results quite different over the lake, or were they quite constant? We didn't actually test because...
For those reasons. So yeah, to answer that question, it was reported by a resident who was astute enough to see that there was a problem, called us actually. I asked her to report to the hotline, and then we started dialogue with the hotline. And unfortunately, that's when 11 blooms happened in Haliburton County. And because ours was frankly limited to 10 square meters, and it was mostly in a couple meters squared, so very, very small. And it was a time when it was dead calm, and I went out each day. I took a sample, and there was nothing else anywhere around the lake. I walked 500 meters either way on the shoreline, talked to a lot of people on Little K, and no one had seen anything else. So partially to answer your question, we don't think it was extensive at all. And we did not submit a sample. We did, as I said, we did submit photographs to Chris Johnson at MECP, and to Ontario Tech, to a prof there who happens to be on our lake. And she confirmed, yeah, looks like blue-green to me. And that was my question to Liz. That report, would it be formally recognized? Probably not. It's recognized in Haliburton County among the lake associations because we've been having a panel, and several other ones happen that way. And just to extend on that question, there is one lake in Haliburton County who had a blue-green algae bloom, which was about 250 meters long and about 75 meters wide. In the end of November, it stayed till ice in, and the algae froze into the ice, and it was still resident in the spring, and it has since dispersed. And it did come back the following year, and the subsequent year, 2021, they did not have a problem. So. Again, as Liz said, it isn't going to happen all the time, but it could happen when Winona Lake was the lake.
One other thing about the testing is when this bloom occurred on Little K, it was my understanding that the process for testing a blue-green algae bloom is not super straightforward. It's not just dipping your bucket in the water and collecting it. So that was why we did not have the correct procedures to actually scoop a proper test. Jim, you can back me up.
No, that's absolutely correct. And so we aren't at the level of maturity yet where we have test kits if there are blue-green algae blooms that we can test them. The MECP insists that their folks come out and do the sampling. So yeah. I think we will evolve to that point someday, but we're not there yet. We're keeping in touch.
Okay. And I have one last question here. Is there any correlation between the measures to eliminate acid rain with the rise of algae blooms?
Yeah, actually there's some new research coming out about that kind of hypothesizing that maybe the lake's recovering from acidification could be the cause of some increases in blue-green algal blooms. I'm not an expert in that field, but there actually is some research going on into that right now.
Okay, and that was the last question that we have.
Okay, and we are right at 8 o'clock, and I have one more slide to close, and then I will thank Liz. Can we see that?
Mm-hmm.
Okay. So in closing, all is not lost. We've actually just initiated a pilot program with 10 lakes in Haliburton County looking at long-term water quality monitoring. In many other places in the province, there's either a watershed council or a conservation authority that actually looks after the health of a region or a watershed. We just don't have that in Haliburton County. So this is part of ULinks and Woodlands and Waterways EcoWatch. I happen to be on the board and the steering committee for WWEW. And these 10 lakes over the next couple of years will be doing a testing pilot, which actually initiates long-term water quality testing. Now, these folks are all in because they've been doing a little bit, but we have 600 lakes in Haliburton County, and we have about 50 lake associations. that represent not all the lakes, but many of those lakes. And the opportunity will be to move that out to the other lakes and it'll be citizen scientist based for now. So, and that has been supported by Halberton County Development, Molson Foundation, the federal government, and Trent University. So stay tuned to that. There'll be a little more of that in the future. And I just really like to thank Liz tonight for a fantastic job, giving us an overview of cyanobacteria and blue-green algae. Really great, and bringing to our attention the things that we can do as waterfront property owners to minimize the risk. Well, thanks very much, and have a great summer. We'll see you on the lake.
Thanks so much for having me.
Yes.
Thank you. And Jim, thanks for facilitating, and thank you everybody for joining us tonight at our first What's Up Kennisis webinar.
Yes, thank you.
Have a great summer.
Have a good
Have a
evening.
good night.
Yeah, thanks everybody.